Good morning, everybody, and welcome to this week's edition of Micro Seminar. I'm very pleased to introduce Dr. Olga Sokolovskaya, who is a very recent PhD grad, well, not that recent, a one-year recent PhD grad from the Plant Microbial Biology Department at UC Berkeley. She has just begun a postdoc at Genentech. But prior to that, she did her PhD with Dr. Michitaga at UC Berkeley and an undergrad at Barnard College, which is part of Columbia University in New York City. And so today she's going to be telling us a bit about B12 and some really cool things about that in the microbial world. And so without any further ado, I'll turn it over to Dr. Sokolovskaya.
Okay, thanks, Cameron. Let me share my screen here. Okay, I should have asked, can you see my cursor?
Yes, I can.
Okay, great. All right, so thank you, Cameron, for introducing me and accommodating me into this seminar series, and thanks everyone who's tuning in. Today, I'm going to talk about the work that I did in Michitaga's lab in the Plant and Microbial Biology Department at UC Berkeley during my PhD and in a short postdoc afterwards. So before I get into the details here, I wanted to give what I think is an interesting historical framework for my projects. So I think probably many of you have heard of B12 or vitamin B12 in the context of human health. B12 is an essential micronutrient for humans, and most of us probably get enough B12 in our food. If not, we can buy supplements at any pharmacy. But before B12 was discovered, anemias due to B12 deficiency were actually a major medical concern. And in fact, one of the anemias was named pernicious anemia because it inevitably resulted in death. And this was the case until 1926, when physicians Minot and Murphy discovered that patients who were fed large volumes of raw liver on a daily basis, as disgusting as that is, those patients could actually be cured of pernicious anemia. And so this discovery led to a search for the therapeutic component of liver. And that was isolated about 2 decades later in 1948 and named vitamin B12. Now, one of the often overlooked contributions to this search for vitamin B12 was made by a microbiologist named Mary Shorb. Mary Shorb was studying something totally unrelated. She was studying the nutritional requirements of a bacterium called Lactobacillus lactis. But she found that the same liver fractions that were curing pernicious anemia patients were also an essential component of her bacterial growth medium. And this discovery was actually very useful for the scientists who were trying to isolate B12 because it allowed them to test different liver fractions using bacterial growth assays rather than testing those extracts on human patients. And so in this way, Mary Shorb expedited the discovery of B12, and that helped open up this huge field of B12 in human health. And this field is kind of marked by the determination of the chemical structure of B12, and that was in 1956. And since then, there have been many, many decades of work on molecular roles of B12 inside of human cells, how B12 is absorbed into the body and trafficked within cells, and also a lot of work on genetic disorders that are associated with B12 metabolism. So obviously all of this work is very important, but what I wanted to point out is that Mary Shorb's discovery that bacteria also require B12 opened up another field of research of B12 in microbial biology. And I'm going to move away from this timeline to introduce B12 in microbial biology, because this is really the context of a lot of my thesis work. But I will come back to these timelines because I want to make the point that I think work about B12 in human health has been mostly parallel to work about B12 in microbial biology. And the second project I want to tell you about today ended up kind of bridging these two fields. So what is B12? B12, which is also known as cobalamin, is an enzyme cofactor, and this is the chemical structure here. It's characterized by a corrin ring, which I am pointing out here with my cursor. And this corrin ring is related to other porphyrins like heme and chlorophyll, but it's a little bit different. And instead of centering an ion of iron or magnesium, a corrin ring here is centering an ion of cobalt. So this cobalt has an upper ligand, which here is R, and this is the chemically reactive part of B12. It's the part that directly participates in chemical reactions. And R can actually vary depending on the nature of the reaction B12 is involved in. So, for example, R can be a methyl group, in which case B12 can participate in methylation type chemistry. R can be this 5' deoxyadenylyl group, and this form of B12 can actually form a radical that can then initiate radical reactions. And the cobalt itself can react with a substrate. And so B12 is actually very versatile chemically. And perhaps this versatility is why B12 has been harnessed by enzymes involved in a lot of different biological processes. There are over 15 different enzyme families that use B12. And in humans, there are only two enzymes that use B12. One is involved in carbon metabolism, and the other is involved in methionine synthesis. But in bacteria, B12 is used for these and many other metabolisms. So bacteria also have B12-dependent enzymes in nucleotide synthesis and nitrogen metabolism, and also kind of specialized processes like organohalide respiration or antibiotic synthesis. And to give you an idea of just how widespread B12 use is in the bacterial domain of life, I wanted to show this summary of some data that was collected by Amanda Shelton, a former member of the Taga lab. Amanda did a bioinformatic analysis of about 1,000 bacterial genomes, and she found that over 80% of these genomes encode at least one B12-dependent enzyme. So what this implies is that the vast majority of bacteria either require or at least can use B12. Now, B12 is only produced by some bacteria and some archaea. And what's interesting is that less than half of the bacteria that use B12 produce it. And this is something that makes B12 kind of interesting on an ecological level, and I'll get back to that a little bit later. But right now, sticking with the structure, I want to introduce you to what will be the star of the show today, and that is a part of B12 that's called the lower ligand. And in B12, the lower ligand is 5,6-dimethyl benzimidazole. That's just the chemical name of this lower ligand. But what's very curious is that there are B12 analogs that have different lower ligands. So for example, pseudo B12 is an analog of B12 that has adenine as the lower ligand instead of dimethylbenzimidazole. And pseudo B12 is also an active cofactor. It has the same reactive moieties as B12 does. And interestingly, we have evidence that in the human gut, pseudo B12 can be significantly more abundant than B12 itself. So this is actually a physiologically relevant analog. And there are actually over a dozen of these B12 analogs that have been either isolated from natural environments or purified from bacterial cultures. And they have all these different lower ligands that I've shown here. And collectively, these cofactors are called Cobamides. So Cobamides have lower ligands that fall into these three major chemical classes you can see here: benzimidazoles, purines, and phenolics. And I think this kind of structural diversity in a single cofactor is very unique to Cobamides, and this is something that made me interested in this cofactor when I first started my PhD. So what the Taga lab and many other labs have done is we've grown different bacteria that we're interested in and provided different Cobamides. And what we see is that in many cases, bacteria can grow with more than one Cobamide, but there are often differences in growth or metabolism that we can observe depending on which Cobamide we add. So based on this, we know that these different cofactor forms are actually functionally distinct. And the functional diversity of this cofactor class makes them interesting on a variety of levels. This is a figure that comes from a review that I recently co-authored, and basically it illustrates different levels at which Cobamides are studied. And so what I'm going to focus on today actually is kind of on a biochemical level, the question of how structural diversity in the lower ligand region of B12 actually affects the biochemistry of this cofactor. How does it affect enzyme function? And in turn, how does that impact bacterial growth? So that's what I'm going to be talking about today. But I also want to mention that bigger picture, we're also really interested in Cobamides because, as I mentioned, most bacteria that use Cobamides don't produce them. So we know that Cobamide producers must be sharing Cobamides with other bacteria. So that's illustrated kind of in this co-culture cartoon where one organism is sharing B12 with another. And if we look even more into complex communities with many members, we think that we have bioinformatic evidence that Cobamides are being widely shared. And we also know that the different forms are functionally distinct. And so we think that these are actually really interesting cofactors to use as a model for studying complex microbial interactions. And so if that's something that's interesting to you, I would point you to this review. I'm not going to talk about that much more today. So with that introduction, here is an outline of what I hope to talk about. So first, I want to tell you about a project that I did to try to understand why these different Cobamides affect bacterial growth in different ways. And I'm going to tell you about a study that I did with the bacterium Cynorhizobium mellilodi. And then second, I want to get back to the human health aspect of this and ask whether human B12 dependent enzymes can use cofactors other than B12. Okay, so here we go. Cynarhizobium mellilodi is a plant root associated bacterium. It colonizes plant roots in these little nodules that are magnified here in this bottom panel. And Cynarhizobium has been kind of a model organism in the B12 field because it is a producer of B12. It has 3 B12 dependent enzymes, and B12 is essential for its growth. And so one of the B12 dependent enzymes in Cynarhizobium is called methylmalonyl-CoA mutase, or MCM. And this is the enzyme I'm going to be talking about today. So MCM catalyzes the conversion of methylmalonyl-CoA to succinyl-CoA. And this reaction is part of a pretty central metabolic pathway. A variety of compounds are broken down through propionyl-CoA and then fed into the TCA cycle by this pathway. So MCM activity is important for probably a variety of different processes, but in the lab, Nietzsche actually figured out that one way to get Cynarhizobium to grow using this pathway is to feed it isoleucine and valine as carbon sources. These are branched amino acids, and they're broken down through this intermediate propionyl-CoA into the citric acid. So what I did, and this was together with Kenny Mock in the lab, was I grew Cynarhizobium in MCM-dependent conditions, so on isoleucine valine, and I actually disrupted the B12 biosynthesis pathway that exists in the organism. And then I added B12 to the growth medium. And so what I'm showing here is not a growth curve, it's more like a dose response curve where I'm adding different concentrations of B12 and monitoring the density that's reached by the culture. And so this growth curve is characterized by an EC50, which is the concentration of B12 that's required for half maximal growth. So here, Cynarhizobium is requiring about 5 nanomolar B12 in the medium to grow. So now I was curious how this organism would respond to other Cobamides. So I added 2 different benzimidazole Cobamides in purple and in pink. And what you can see is dose response curves shift to the right, and this corresponds to an increase in the EC50 values, almost 2 orders of magnitude. So clearly, this organism is responding differently to different Cobamides as we expected. And this was also true when we grew Cynarhizobium with pyrenyl Cobamides. They all had EC50 values that were higher than B12 and varied between the different pyrenyl Cobamides. And the same was true for phenolyl Cobamides. They both had EC50 values 2 orders of magnitude higher than B12. So what I wanted to know was, why is this bacterium requiring much higher concentrations of other Cobamides than it is of B12? And probably the most straightforward explanation would have been, oh, maybe it's just not taking up these other Cobamides as efficiently. But we actually extracted the Cobamides out of cells, and between the 3 classes, we didn't see major differences in the amount of intracellular Cobamide. So that didn't seem to really explain these results. So I hypothesized that perhaps the reason that we were seeing differences in growth is that these Cobamides differed in their ability to support MCM activity, since after all, we are growing the organism in MCM-dependent conditions. So here is a crystal structure of MCM. This is actually human ortholog of MCM. And in gray is the enzyme itself, and in blue is B12. And the part that I've put in the box is the lower ligand, which is the structurally variable region. And if you kind of zoom into that area right here, what you can see is the lower ligand, which is in blue, is really surrounded by protein residues. So it's bound within a binding pocket in the protein. And this observation leads to one hypothesis, which is that perhaps differences in the structure here would change the ability of the cofactor to bind to the protein. So that is one hypothesis that I wanted to test. Another hypothesis that I wanted to test, and I wanted to kind of do it separately, was to test whether the lower ligand could actually change the catalytic properties of MCM. So to test these hypotheses, I went ahead and heterologously expressed Cynorhizobium MCM, purified it, and the next step was actually to produce all of the Cobamides that I had to use, because the only one that you can buy is B12. And the way we produce these Cobamides in the lab is by a process called guided biosynthesis. We basically grow up very large volumes of bacteria that make Cobamides, and we add high concentrations of the lower ligand that we want the bacteria to incorporate. And so in some cases, we can get efficient incorporation of other lower ligands. And then what we do is we collect the cells, break them open, and purify out the Cobamides. And what I wanted to show here on this photograph here is that one of the nice things about working with Cobamides is that they're colored. They're actually red. And so in this chromatographic separation here, I pretty much know that what I'm looking for is probably in this red band here. So I purify these compounds, then this is an example of something that I might end up with. And last step is that I do a chemical modification specifically to install the 5' deoxyadenylyl upper ligand, which is a catalytic upper ligand that's used by MCM. And something that's notable about this form is that it's actually sensitive to light. So all of the experiments that I'm going to show you, all of the in vitro experiments, were actually performed in the dark. Something else I wanted to point out that's kind of useful is that these cofactors have really distinctive absorbance spectra. So here there are two spectra. The black one corresponds to this form. The blue one corresponds to this form. So you can see as you change the structure of the cofactor, you see big differences in the spectra, and that's really a useful tool for studying these molecules. So that's just a little bit about what it's like to do this work. Okay, so the first thing I wanted to do was to characterize how the lower ligand impacts the binding of MCM to Cobamides. And specifically, I wanted to determine a KD, or an equilibrium dissociation constant, for each Cobamide, and that describes the binding affinity. And something I took advantage of to do this is the fact that Cobamide binding to MCM actually quenches the intrinsic fluorescence of MCM. It quenches tryptophan fluorescence. And so in this fake data plot here, you can see that if you add more and more ligand, in this case Cobamide, the fluorescence of MCM decreases. And this decrease can be fit to a binding equation that reveals the KD. So here is a binding experiment with purified Cynorhizobium mellilodi, MCM, and B12. And again, B12 is the native cobamide produced by this bacterium, so definitely expected it to bind to the enzyme. So luckily here you can see we have a decrease in protein fluorescence as we add more and more B12. And that corresponds to a KD of about 0.03 micromolar. So now instead of adding B12, I added a cobamide that differs by one methyl group here, by two methyl groups here, and one that has a hydroxyl group. And you can see whether you look at the numbers here or just look at the graph, you can see that these cobamides have different binding affinities for MCM. So clearly, even these small changes in the lower ligand structure are affecting the ability of these cofactors to bind MCM. So these are all benzimidazole cobamides here. What about cobamides from other structural classes? So here are two phenol cobamides, and you can see that even though they're from a different structural class, they actually bind MCM quite well with affinities, same as or close to that of B12. In contrast to that, both of the purinol cobamides that I tested did not bind the enzyme that well. So this orange one here had kind of an intermediate affinity, and the red one, which is pseudo B12, which I introduced earlier, pseudo B12 didn't really bind at all at micromolar concentrations. So again, this result kind of reiterates the point that cobamides with different lower ligands vary in their ability to bind MCM. Now, the question is, can these differences explain the differences in growth, the differences in EC50 that we observed in vivo? To get at that, I made this relational graphic where I plotted the in vitro KD on the x-axis and the EC50, which was from the in vivo experiments, on the y-axis. And I'll point out here that the x-axis does have a break right here. Some of the cobamides had KD values that were too high to measure. So I don't know exactly where those points are on the x-axis, but I know that they're higher than 10 micromolar or so. So what you can see in this plot is that for the benzimidazole cobamides in blue and for the purinol cobamides in orange, there does seem to be a trend where the higher the KD value, the higher the EC50. So in these cases, it seems like the KD could actually explain potentially the differences in growth. An exception to this are the phenol cobamides. They have low KD values, but they're still required at high concentrations for growth. And so binding does not explain why cyanurhizobium requires high concentrations of these cobamides. So as I mentioned before, I also wanted to test whether lower ligand structure impacts catalysis by MCM. And the way I did this was I used a coupled assay. So let me explain that. So basically what I did was I combined MCM with different cobamides, and I added those cobamides in excess so that all the enzymes would be bound with cobamide. And then I added the substrate, methylmalonyl-CoA, and I wanted to monitor the rate of formation of the product succinyl-CoA. Succinyl-CoA is a little bit tricky to detect. And so what I did was I coupled the activity of MCM to this enzyme thiokinase. And what thiokinase does is it releases CoA from succinyl-CoA, and CoA then reacts with another reagent I added that's abbreviated DTNB. The details don't matter that much. The point is that this reaction liberates a compound that has a strong absorbance at 412 nanometers. So this compound is yellow. And so basically I can monitor the formation of this compound as an indirect measure of the formation of succinyl-CoA. So here are the data that I got from that assay. So what I'm showing on the left is a classic Michaelis-Menten kinetic curve of the enzyme with different Cobamides. So what this is a plot of the reaction rate as a function of the amount of substrate that was added. And so, I mean, if you just look at it, what you can see is the enzyme is active with all of the 4 Cobamides that I tested. There's no Cobamide that, inhibits the activity of the enzyme. So that's pretty clear. But there are a couple of parameters that are typically measured about these plots. One is kind of the maximum rate that's achieved by the enzyme. And that maximum rate is used to calculate a kcat, or what's known as a turnover number, the number of molecules the enzyme turns over per second. And so you can see that B12 has the highest turnover, about 26 per second. And the other Cobamides, again, support activity, but there are maybe 2 to 3-fold lower turnover. I do think it's pretty notable that even, from here to here, a difference of 1 methyl group was enough to change the catalytic property of the enzyme. So I think that's pretty interesting. So 2 to 3-fold differences in kcat. Another parameter of these curves is the Km, and that's the concentration of substrate that's required for half maximal activity. And the Km you can see for all 4 Cobamides is pretty similar. So this Km isn't really affected by Cobamide structure. And the third number that's listed here is a ratio of the 2, the kcat Km. This is used as a measure of catalytic efficiency. And as you may expect based on the numbers I just told you, B12 has the highest kcat Km, and the other Cobamides have 2 to 3-fold lower activity. So clearly, lower ligand structure is affecting enzyme catalysis, but not by much. We're seeing differences of 2 to 3-fold. So I want to now come back to this growth phenotype that I started with, that the phenolcobamides are required at concentrations 100-fold higher than B12. This was not explained by binding, since these cobamides had similar affinities as B12. And I also don't think it's really explained by activity, because I only saw differences in activity about 2 to 3-fold with these cobamides. So I think there must be some other factor in the cell that is causing these cobamides to be required at such high concentrations. So here are the different molecular factors that I think can contribute to cobamide-dependent growth. So, these little circles represent different cobamides. So one thing is, we have evidence that cobamide import is affected by cobamide structure. There are cases where bacteria will preferentially import one cobamide over another. But as I mentioned, in this case, it didn't seem like there were major differences in the amounts of cobamides that were getting into cells. So I don't think this is a major determinant in the case of Cyanorrhizobia mellilodi. Something I didn't mention is cobamide adenosylation. So when I was doing the in vitro experiments, I chemically installed the reactive upper ligand, the adenosyl upper ligand. But in the bacterial growth assays, the bacteria have to do that reaction. And so it's possible that these phenolcobamides are inefficiently adenosylated. And that could be something that we would have to test, but that could be something that explains the fact that they're required at such high concentrations. For the other 2 classes, the benzimidazole cobamides and the pyrenol cobamides, it did seem like the differences in binding to MCM could explain the differences in growth, as you could see in that relational graphic. And finally, I did see that lower ligand structure could affect the chemistry of the enzyme. And so that probably contributes to growth as well. So basically, we think that by understanding these different factors in the cell, we can get a better sense of basically what determines cobamide-dependent growth, and that this could be really useful in the future as we start to study the roles of these cobamides in more complex systems, such as in co-cultures or in microbial communities. So that's kind of our goal with this work. So just to finish off this section, I also wanted to ask how MCM selectivity would differ between organisms that use different cobamides or make different cobamides. So I also expressed MCM from E. coli. E. coli doesn't actually make cobamides de novo, but it can take a late cobamide precursor and it can itself attach a lower ligand. So it attaches to methyladenine here. And Veillonella parvula makes a phenol cobamide. And so I also expressed these two MCMs and I assayed their binding to 6 different cobamides. And here I've grayed out a lot of data. I just colored the curve corresponding to the native cobamide of each organism. And I did that just because I wanted to point out that in each case, the native cobamide has high affinity for the enzyme. So this kind of makes sense and suggests perhaps that cobamide biosynthesis has co-evolved with cobamide selectivity in MCM. So that's the first part. To finish here, I want to get back to the human aspect of B12. So I pointed out that work on B12 in human health has kind of gone in parallel to work on B12 in microbes. And part of the reason I say that is there really hasn't been that much investigation of how these different forms of B12 produced by bacteria and archaea impact humans. And part of the reason I think this hasn't been explored is that B12 uptake by humans is thought to be highly selective. So we have diverse cobamides in our diets, but in the small intestine, the cobamides have to be bound by a protein called intrinsic factor in order to be taken up into our bodies. And intrinsic factor binds B12 very, very tightly. It has been characterized to have a KD for B12 of about 5 femtomolar, so very tightly binding. And studies have shown that intrinsic factor binds to other cobamides, like these are 2 pyrenyl cobamides, pseudo B12 and factor A. Those are bound by intrinsic factor with much lower affinity. So the KD for those is about 3 nanomolar. And the KD of intrinsic factor for cobinamide, which is a B12 precursor, is even higher, about 40 nanomolar. So based on this, people have kind of said, well, intrinsic factor only binds B12, so no other cobamides get into cells. I would challenge that with 2 points. One point being that there are, I showed you there are over a dozen different cobamides, and many of them are very similar in structure to B12. And there actually is some evidence that those cobamides are bound more tightly by intrinsic factor. So it's possible that those cobamides would get into cells. That's one point. And another idea is, an affinity of 3 nanomolar is actually a pretty decent binding affinity. And perhaps it could be the case that if someone were to ingest foods high in pseudo B12, which there are foods that are high in pseudo B12, perhaps those pseudo B12 could reach a concentration of, in the nanomolar range in the gut, and therefore be bound by intrinsic factor and absorbed into the body. So I think those points kind of warrant an investigation of how these cobamides would impact human B12 dependent enzymes. On top of that, around 1980, I found a few studies that actually report non-cobalamin B12 analogs in human tissues and also in animal tissues. So there's some evidence at the time, researchers didn't know what these analogs were, but there is some evidence that there are B12 analogs of some sort in our tissues. So I think based on this, I really wanted to ask, how do these other cobamides impact B12 dependent enzymes in humans? So as I mentioned, there are two B12 dependent enzymes in humans. One is methionine synthase. I'm not going to talk about it. The other is MCM, which is the enzyme I had been studying in bacteria. And in humans, this is a mitochondrial enzyme, and it's part of a very similar pathway as it is in bacteria. So I went ahead and heterologously expressed human MCM, purified it, and characterized it in the same way that I characterized the bacterial enzymes. And I won't show the details of my characterization. I'll just show one experiment that summarizes the main point. So what I'm showing here is a plot of the specific activity of the enzyme. It's just rate of the enzyme reaction normalized to the amount of the enzyme in the reaction. And what I've done here is I've provided the enzyme either B12A, which is A here, or these other cobamides, which are B through I. And so you can see it's active with B12. And maybe as a negative control of sorts, I'll point out that cobinamide, which is a B12 precursor, is known not to support MCM activity. So here with I, we don't see MCM activity. You can see that B, C, G, and H all support human MCM activity. B and C are two benzimidazole cobamides, and G and H are the phenol cobamides. So clearly, human MCM is active not just with B12, but with other cobamides. Now, there are variants of MCM in the human population that have single amino acid substitutions that result in a metabolic disorder called methylmalonic aciduria. And in 2014, Patrick Forney and Sean Froese did a study where they investigated the biochemical defects that are associated with some of these substitutions. And a subset of the MCM variants that they investigated, including the 6 that I've pointed out here, actually seemed to have a decreased apparent affinity for B12. And when I read this, I was intrigued because I thought, what if these variants, while they have decreased affinity for B12, what if they have an increased affinity for a different cobamide, or even an unchanged affinity for a different cobamide? If that were the case, perhaps a different cobamide could be used as a therapy for this metabolic disorder. So I reached out to Wyatt Yu at the University of Oxford and Matthias Baumgartner at University Children's Hospital in Zurich. And we collaborated to study how these different MCM variants respond to different cobamides. So what I did was I basically took my library, if you will, of cobamides that I had generated over the course of my PhD. And so that consists of 16 lower ligands shown here. And I basically screened these Cobamides for their ability to improve the activity of these human MCM variants. So here's an example with wild type enzyme of the data that I'll show. This is again the specific activity with the enzyme, and the bars A through P just correspond to these different Cobamides. So here is the activity of 6 disease-associated variants with B12. And if you compare the y-axis values, you'll see that all of the variants have much lower activity than wild type, and that was expected. But what I hoped is that maybe a different Cobamide would boost the activity above A. And this was kind of a long shot, but it seemed worth a try. Unfortunately, that's not what I observed. All of the other Cobamides either supported activity comparably to B12 or to a lesser extent. So it doesn't seem like any of these Cobamides are promising therapy-wise for these 6 MCM variants. But what was interesting was that these mutants all were active with Cobamides other than B12, just like wild type enzyme. So G and P support activity in many of these mutants. So again, to summarize that, it seems like both the wild type MCM and even disease-associated MCM variants can use Cobamides other than B12. And I think this is useful to know in 2 respects. One is it implies that if Cobamides do get into cells, Cobamides other than B12, perhaps they would not necessarily be detrimental to our B12 biochemistry. So that's one point. And another point to consider is that, you know, there are people who don't eat meat, and meat is our major source of B12. And there are also populations that don't really have access to meat. And so it would be very useful if people who don't consume meat could consume foods that are rich in other Cobamides and be able for those Cobamides to be able to support their B12 metabolism. So it's just something that I think is useful to think about. I think one open question related to this topic that is very important is whether these Cobamides ingested, you know, through orally can actually reach MCM in our bodies. And the reason that's a question is because B12 absorption and trafficking is actually very complex. So this is a complex diagram. The details aren't important, but it's basically showing how B12 in red goes from the stomach into the ileum, into the blood, and then within cells, how it's moved around until it reaches the MCM active site or the methionine synthase active site. And everything that's boxed is a protein that directly interacts with B12. And we don't know how any of these proteins interact with any of the other Cobamides. So I think there's still really a lot to learn about the relevance of Cobamides in human health and how human enzymes or proteins respond to different Cobamides. So just to end, I want to show you one thing that I think is really remarkable that came out from this work. And you can see it if you directly compare side by side the Cobamide binding profile of Cynorhizobium MCM and human MCM. You can see that they have extremely similar Cobamide binding properties. And I think that's really striking given that one is a bacterium and the other is a human enzyme. And this observation caused us to speculate maybe about the evolution of this enzyme. So here I'm showing scheme that demonstrates the evolution of archaea, eukaryotes, and bacteria from the last universal common ancestor. And what I wanted to illustrate is the fact that we believe that the human mitochondrion evolved from a bacterial ancestor that is thought to be most closely related to alpha proteobacteria. Given that alpha proteobacteria and human mitochondria share a relatively recent common ancestor, I think it's really interesting that I observed biochemical similarities between an enzyme from Cynorhizobium and an enzyme from human mitochondria, and that the selectivity of those enzymes was different from MCM in E. coli and MCM in Veillonella parvula, which are organisms that share a more distant common ancestor. And so this finding got us really interested in the evolution of Cobamides selectivity, and it's something that we think will teach us potentially about kind of the molecular mechanisms by which this selectivity has evolved. So that's something that the lab will pursue in the future. So with that, I just want to acknowledge the contributions people have made to the work I told you about today. Nietzsche and current and past members of the Taga Lab have contributed a lot to this work, and I wanted to point out specifically Kenny, Jong, Catherine, and Anna, who contributed directly to the project that I told you about today. The growth curves, the bacterial growth curves that you saw were done by Kenny. I wanted to acknowledge our collaborators at the University of Oxford and University Children's Hospital in Zurich. Again, this was the collaboration on the disease-associated human MCM variants, and that was a really nice collaboration. And that's it. I would be happy to answer any questions. And I will stop the share here.
Great. Well, thank you very much. That was really stimulating, and I had a thought at some point about the connection with mitochondria, and that was really cool. That I was like, wow, that's really out there.
Yeah.
I'm glad that you put that up, because it's really compelling to think about the alpha proteobacterial ancestry and the relationship to the human kinetics. So I have a couple questions, but there is one from the audience, and then I can ask mine, and we're on a little bit of a time delay, so it's good for me to have some fillers. But the one question is just in terms of like using terminology. Novita Kurniawati, I hope I pronounced that right, is in Indonesia and was wanting to understand a little bit more about why you were using the term analog versus synthetic for Cobamides.
I used versus synthetic. So I will point out that all of these different Cobamides are, at least all the ones that I talked about today, are natural in the sense that they can all be found in nature. They're not ones that are synthesized in the lab. I use the term analogs just in its common meaning to say that these compounds are analogous to B12.
Yeah, so a synthetic compound would imply that that's a chemical creation by a human, as opposed to I guess it could be biosynthetic, but it's something that's engineered as opposed to just something that's naturally different.
Perhaps. And we did actually design in our work some unnatural Cobamides that we produced as well in the lab. So maybe those could be referred to as synthetic. I'm not sure if that's what was meant in the question, but. Yeah,
okay. Well, thanks. Coming back to that, I was curious. I don't know if I missed this, but what were the organisms or organism that you used to do all the synthesis of the Cobamides?
Yeah, so we have a few favorite organisms to use, and they're favorites because they produce relatively large amounts of cobamide. It's all relative because it's not huge amounts, but we use Sporamusa ovada, which is a firmicute, and that we use Sporamusa to produce phenolcobamides and some benzimidazole cobamides, but Sporamusa doesn't attach the purines. It just won't attach them in the cobamides. So we use a different organism right now called Propionibacterium acidipropionici, which can attach the purinal lower ligands more efficiently. So kind of depending, there are other organisms we've used, and depending which cobamide we want to produce, we will choose the organism that we think might best produce that cobamide. And sometimes it's something we have to troubleshoot a little bit.
Yeah. Well, I mean, that's fascinating in and of itself that you have these different cobamides analog producing differences among different taxa. Is that process well understood in terms of like what are the pathways and why, like for example, the firmicute won't attach the phenolic compounds and why a propionibacterium would? I mean, is that all like pretty well tracked out, or is that also an area of investigation?
It's an interesting area that we know some amount about. Just to clarify, maybe I said it's the purinal cobamides that are not attached by Sporamusa and attached by the propion.
I'm sure.
I don't know.
I'm sure I got
it doesn't
that
matter.
myself.
But so this is actually an interesting area of work that has been done. There's an enzyme involved in attaching lower ligands called COB-T, and our lab has actually published a little bit on the selectivity of COB-T, so selective attachment of lower ligands. And we know a little bit about it. We know that there's a another enzyme related to COB-T called ARSA-B, and that's the one that we know attaches the phenolcobamide. So we know that only an organism that has ARSA-B will be able to attach phenolcobamides. So that's something we know, but we don't know enough to like always be able to predict what lower ligands will be attached by an organism.
Yeah, cool, interesting. All right, we have another question from John Moreau. Hi, John. It's notable that mercury methylation is absent from the human gut. Microbial methylation genes are rare or absent. Could there be a relationship to B12 or cobamide availability, i.e. affinities?
Okay, so yes, B12 is involved in a process called mercury methylation. I don't know a whole lot about it. I've heard of mercury methylation kind of in the environment, but I've never heard of it in the context of the human gut. Does the question say that mercury methylation is absent in the human gut? And what is the second part of the question?
Could there be a relationship to B12 or cobamide availability?
To the availability. I'm not sure to the availability. I mean, I think the concentrations, I don't think that cobamides are particularly scarce in the human gut, at least that's not what we think based on concentrations that have been estimated. So I would guess it's more related to the presence or absence of the mercury methylation genes rather than the availability of cobamides, if that answers the question.
Okay. I'll see if he wants to follow up. Just in the meantime, I wanted to, you had this conundrum at the end where you had orders of magnitude differences in growth, but only a few fold differences in activity.
Mm-hmm.
I was wondering if there's anything about the growth you were doing, the growth assays, that might also be B12 dependent besides the MCM activity itself that could account for that? I mean, is there some other activity that might be suffering from the B12 analogs?
That's a good question. So the only, so there are two other enzymes inside rhizobium that use B12, and we actually deleted or disrupted those genes so that those two specific enzymes wouldn't impact our measurements. So besides those enzymes, I can't think of anything obvious that might be kind of interfering with our measurement. But if I think of something, I'll let you know.
Sure. Okay, yeah. Well, thank you very much. There's no further questions in the chat, but there may be other ones later on, so definitely check back. And thank you for another fascinating micro seminar. We really appreciate your time.
Yeah, thank you.
Okay.